Okay, so it's my great pleasure to introduce our next invited keynote speaker. This is Dr. Alisa Manning from the Broad Institute of MIT Harvard. She's an assistant professor and associate member of the program in medical population genetics and is assistant investigator at the Clinical Translational Epidemiology Unit at Nashville. She's a statistical geneticist by training with a focus on glycemic traits and a relatively new focus on integrative analysis of whole genome sequencing data. Her lab studies the molecular and mechanistic basics of insulin resistance and obesity with the overarching goal of trying to understand the relationship between genes, environment, and disease, foster stimulating and multiplicity research program to advance on these topics. So thank you very much, Alisa. We're really excited about it.
Thank you. All right, the title of my talk, 10 Years of Genetic Epidemiology in the Cloud, How Open Architecture Has Won the Day. You know, we're gonna get through all these tough words. My outline is, I'm going to give a very quick introduction to genetic epidemiology, an introduction to my research and how we work, and then give you a history of from my perspective, just, you know, from the work that I have done over the last almost 20 years, how I've moved towards open architecture in my genetic epidemiology studies. And then I'm going to provide some examples of open architecture solving problems in genetic epidemiology research. So open architecture basically here is the cool stuff we can do by this NIH-funded platforms that we have. AnVIL is like one of the best examples of open architecture. All right, so what is genetic epidemiology? The next two slides, I'll go through and take a couple resources, quotes from these papers that really set forth what the field is. So genetic epidemiology is the study of the etiology, distribution, and control of disease in groups of relatives and of inherited causes of diseases in populations. Sourced from genetics and epidemiology. Here, we're going to study defined populations in defined ways. This is what makes genetic epidemiology a little different than computational biology, data analysis, or bioinformatics. We are thinking about study design and effects. And you know, in a group of people with rich phenotype data, there's a whole lot that you can study and a whole lot of effects that you can seek out. So we're trying to quantify the risk for effects estimates associated with genetic variation, and that is a prerequisite for assessing the use of this new knowledge in medicine. Another resource paper published in 2016 says that within public health genetics research, genetic epidemiology is hybrid of epidemiological designs and statistical models that explicitly consider both genetic and environmental risk factors for disease. And here we can use the field to improve prediction of individuals at risk, design and implementation of targeted biological interventions, and gain deeper insights into the biology of disease. So genetic epidemiology is more than just looking at Manhattan plots of phenome-wide association studies and regional plots plotted with like the locus tool. I'm putting up here some figures from one of the papers I did at the end of my graduate studies, where we were studying glycemic traits, so fasting glucose or fasting insulin levels in human population cohorts. And I at the places where we saw genetic association, I wanted to explore what different models, what the significance of that variance is with different models, and then what the effect size in different contexts. So we were studying gene-environment interaction with body mass index or obesity as a physiological environment. So when I do my genetic studies, usually I'm trying to incorporate environmental factors, lifestyle factors, with many different types of genetic factors to study traits and diseases. So let's get into how we work. So our team is, our research is meant to study, I say the word, understanding the relationship between genes, environment, and disease. I've got a research team at Mass General Hospital in the Clinical Translational Epidemiology Unit, and in the Broad Institute in a new initiative called the Diabetes Initiative at the Broad Institute. So I've got a great team of academic trainees, most of whom have graduated and are now faculty analysts, and now on the other side of the Broad Institute, I also am participating in the Biodata Catalyst platform, and I'm working with many folks you know in the AnVIL. Kate and Jonathan are right on top of things here in AnVIL, so they're also contributing to a Biodata Catalyst. So what we do, in my research, I am in the area of statistical genetics, genetic epidemiology, and molecular epidemiology, now studying many of the omics factors in population cohorts. I study type 2 diabetes and insulin resistance, and I work on developing and making available methods and software for workflows for doing whole genome sequence association studies, and then methodology, software, and tools for gene-environment interaction models. At the data science platform, as I mentioned, I'm involved in Biodata Catalyst, but then there's a really important effort for interoperability with NHLBI's Biodata Catalyst with NHGRI's AnVIL and other of these platforms we've already been introduced to. The Recover project, we're studying long COVID at the NIH, is part of Biodata Catalyst. We support the Recover data in Biodata Catalyst. And then interoperability projects, one that I'm working on with Mike here, study communicating methods of interoperability for AnVIL and Biodata Catalyst, and then the NIMDH Share portal and the NHLBI Biodata Catalyst. So what's the funding of our work? I've been really fortunate to get funding from a lot of different institutes to do a lot of different types of studies. So from NHLBI, NHGRI, NIDBK, and then the NCPI projects, which I think are funded out of the Office of Data Science Strategy. For NHLBI, I'm involved in doing analysis in Biodata Catalyst in the TopMed study, the TopMed program, and also an active researcher in the CHART consortium. Not only am I part of the infrastructure platform building part, but I really do use that in my research. I'm part of the PRIME consortium, which is funded by NHGRI and uses AnVIL for data analysis. I'm fortunate to have been part of the Accelerating Medicines Partnerships programs for type 2 diabetes and common metabolic diseases funded by the FNIH in partnership with the NIH. And what are the platforms that we use in our work? So I use Biodata Catalyst powered by Terra for workspaces. I use GitHub for code, DocStore for workflows, and then a couple other tools that are a mixture or part of all the infrastructure that is built into these the use of workflows in the Terra platform or AnVIL. And then I also do work in All of Us. I do work in UK Biobank. So it feels like I'm just all in cloud computing and doing my epidemiological research in the cloud. In fact, I barely ever log into our local on-prem high-performance clusters, both at the Broad Institute or MGH. So I really have moved all of my group's research into the cloud. So let me tell you a history of how we got here. Back in my graduate school days, between 2005 and 2011, I was at Boston University using our local cluster. My job prior to starting my PhD program was to build an analysis pipeline for the first genome-wide association studies for the Framingham Heart study. And our pipeline and architecture evolved over the course of 4 or 5 years, where the first type of GWAS that we wanted to do, well, we did it for them. We just wrote the code that ran 100 different genome-wide association studies and all the different trait domains for the Framingham Heart study. Then when the next tranche of data came around, our pipeline evolved so that the investigators could run, they could bring their own phenotype data And so we had all set up all the genotype data for them and trying to make it simple and easy for them to run their own genome association studies. So in this era, I became familiar with open source software. The next era, where I was a postdoctoral research fellow at the Broad Institute. I'm using the local cluster again, and the methodological problem is doing efficient computing with mixed models for our genetic epidemiology studies, where we have whole genome sequence data in cohort studies for the first time. So around 2011, we were fortunate enough to be working in 2 NIDDK-funded projects that were generating whole genome data in studies where we can, cohorts where we could do these phenotypic genome association studies. So we had to figure out how, because previously we had only these genotyping arrays, and the imputation was starting to become something that people did, but we had to find tools, build the tools within our consortium to do the analysis that we need to do. And here, during this period of time, open data became critically important for us to use. And there's a couple examples of sources for open data that we use. And then, more nowadays, moving into the current era, things got pretty exciting. I was fortunate enough to be involved in the TopMed study when whole genome sequence data was being generated for a large number of cohorts. And in 2016, the CHARGE Consortium, which is a group of cohorts that whose leaders got together and thought about, okay, well, how can we put our data together and do analysis together? So our first foray into cloud computing was through a consortium data sharing agreement that allowed data sharing and analysis on the DNA Nexus platform. And then in 2017, the TopMed study was getting going, and we had these cloud computing pilots where the FireCloud platform, which eventually became Terra, was the place where it was my playground. And I had an amazing time talking to these the people building FireCloud and explaining to them how the analysis that I was doing was different than maybe the analysis from the NCI data that they were familiar with. That yes, I wanted to run an analysis a second time because I wanted to change features of that analysis to ask a different question. So the exploration of using FireCloud for my genetic epi studies was really getting up full steam around this period. And then in 2018, 2019, 2020, and online in 2021, the NHGRI and the NHLBI encouraged and provided for this GSP TopMed analysis workshop. And here the cohorts where the investigators involved in generating data, the CCDG data that we've already learned about a little bit today in NHGRI cohorts, and the investigators generating whole genome sequence data in the TopMed cohorts, we got together repeatedly to talk about best practices for doing analyses with these data. In 2020, I was involved in an NCPI interoperability pilot showcasing co-analysis of kids' first data, AnVIL data, and biodata catalyst data. In 2020, Biodata Catalyst threw open the doors, launched their fellows program to fund young investigators doing the research in Biodata Catalyst. And a bunch of things happened between then and now. And in 2024, I just want to highlight that we showcased an interoperability between the NIMDH Share portal and Biodata Catalyst and the NCPI interoperability project that I'm working on right now. So here, the open architecture and platforms really accelerated our research. So we've got workflows where we have our code, registered in DocStore, searchable and findable, and exportable to Terra, and all the different platforms. I'll note that Biodata Catalyst is a collection of platforms, and so we had to learn interoperability and build in interoperability in Biodata Catalyst, which using open standards and established standards really made our work possible. And finally, the data use oversight system Duos that is now available. I'll have a little bit of an example of using that in AnVIL data analysis. Okay, catching up a little bit on time. I'm going to go through a few examples of open architecture solving problems in genetic epidemiological research. The first is if you're a stats person like me, I do have my hands in methods development in the area of genome interaction studies. So I was fortunate to have an R01 funded with my close colleague, Han Chen from the University of Texas at Houston. The title of our project was Methods and Software for Large-Scale Genome Interaction Studies. And we have developed a software suite of tools to enable the expansive genome-environment interaction studies on like millions of people. That's our goal. One of the features of our software tool is our suite. We have our flagship software GEM, but then we have ReGEM and MetaGEM that leverage the summary statistics generated from our the GEM software to do reanalysis and ask different questions about genome-environment interaction effects using summary statistics generated in the more expensive individual level data analysis that GEM provides. So we have a suite of tools where we want to get it out there. We want people to use our tools. So how do we use the current suite of open architecture platforms and tools to enable that? So we want the widespread adoption of our new statistical methods. So we have our GitHub repository where we have our software and the instructions of how to download the software and compile it and all the prerequisites. But then, and then we have our paper and all the stuff that us methods folks want to see. But then we'll use containers. So at one point in time, we put all of our Docker containers in the key.io. So each of our desired uses of our software are represented in a container, and then we have DocStore where we have our workflows. So people can now go and search for GEM or search for our name or large-scale environment interaction studies, and they'll find our organization and find us in DocStore. So the advantage here is that researchers can find and use your workflow right away. There's the great launch with a feature in DocStore to export your a workflow into the environment, the set of environments in which it is that are built for it. In this case, this is the WDL workflow language. So then we've got flexible configuration in Terra. So Jonathan talked a little bit about some of the recent feature developments and future feature developments in Terra. But here, I just want to highlight that you learn how to configure a workflow in Terra. It can take you a long way because you can do this with Biodata Catalyst data, with AnVIL data, and so the platform is just widely used. And you can run the workflow, and here the advantages are you can track details like the timing. Could have thrown up a screenshot of tracking the costs of the workflow, but I like the colors and the timing diagram. And then you can keep track of all your workflow runs. I mean, this has just opened up everything for me because my analyst says, I came across an error. I just can use the tools available to me in Terra to dig in, after 20 years of experience, I've seen every bug on the planet. So like I can like that's what you do. When you're a PI, you're an expert debugger. I can help them because I can directly see the complete configuration of the run of a particular analysis workflow. Like there's no guesswork. And in fact, it's really frustrating when the analysis run is performed in another research environment, and then I ask about, what about, well, just prove it to me. Prove to me how you did it. And there is no record. So being able to maintain the provenance and also the history of analytic workflow runs is really just a great advantage. Okay, then let's get into another problem, variable discovery in NHLBI cohorts, the cohort data hosted in Biodata Catalyst. So here I have a research example. I'm a genetic, I want to design a genetic epidemiology study of heart attack in individuals diagnosed with type 2 diabetes. So in the old days, I was just wanted to explore the data available in different data sets that maybe are these NHLBI top med cohorts. I would have to go to the dbGaP page for each of the studies, search and read about all the data sets and the variables. And dbGaP's a great tool, but sometimes if you have 1,000 variables in a data set, you have to page through each set of 50 to just read what the variable descriptions are. In Biodata Catalyst, we have the Picture platform, where one quick way to get to it, as I'm showing here, is on the Biodata Catalyst page, going to explore BBC data, and just click on the phenotypes button, and you're on the Picture powered by Biodata Catalyst platform, where you can. There we go. Yeah, so here we are on Picture, where I've entered the words heart attack, and I immediately can see of all the data hosted in Biodata Catalyst where these words are appearing. And then, of course, your work to do more clever searching is underway in Biodata Catalyst. But this takes you a long way, because within 30 seconds, I can type heart attack, I can type cardiovascular disease, I can just do a quick search of all the terms I know are most relevant for my question and discover the cohorts and the variables, and then click on the descriptions to get counts of individuals with the different values of this variable. So it's a great a tool for people as they're doing data exploration and building out study designs for their genetic epi studies. All right, then finally, so let's say I want to do an analysis that leveraging Biodata Catalyst data and AnVIL data in the same workspace for the analysis. And so we are really, privileged that, or we have available to us the Terra platform in which we can do this. And so I'm showing a Terra workspace where I want to study a particular disease. I want to study gene-environment interaction effects near genes that have different gene expression levels due to a lifestyle factor. So I want to leverage a data set like GTEx with its broad swath of just that very valuable multiple tissue data to discover a set of genes that I want to go and evaluate in a disease-specific study in another cohort. So, this is kind of a made-up demonstration project, but I'm trying to make the point here is that I want to do all the work in one place. Maybe I only have a single building project set up, or I'm running a research lab, and I want everybody to be using that same workspace so I can easily track the entire analysis as a PI. So to create this workspace, I've used Duos as a mechanism. Researchers can use Duos as a mechanism for requesting data access to AnVIL projects. I would refer to all the documentation available on the AnVIL website. But then, when you have a data access request approved, then you can use the AnVIL data library to export data to a workspace that Duos has an export feature as well. But basically, what we end up with is a workspace that has AnVIL data. I know this is really, really tiny, but the point is that the data model is set up in the expected way. If that's what people who are familiar with AnVIL. But then the biodata catalyst data is also set up in the expected way. It's a different table in the data model. And then, you can just proceed with your analysis. So that's the completion of my talk. I'm happy to answer questions. I feel like I've been everywhere in cloud computing when it comes to genetic epi studies. So come and ask me questions. I'm more than happy to share my experiences with you. So thank you.
All right, thanks. Oh, wow. Oh, thanks, Lisa. I think we may have time for one question.
Hi, I was just wondering, do AnVIL and Terra also keep track of workflow runs, or is that only a biodata catalyst feature?
So many of the, when you see AnVIL workspaces, they're built the same way with the same functionality as Terra workspaces and the biodata catalyst powered by Terra workspaces.
Perfect, thank you.
Thank you. I want to ask you, when we go to the platform, which language we want to prioritize? Do we learn HAIL first, or do we? Or Python or R bioconductor? Which one we want to dig into it?
Well, we can use HAIL in AnVIL, biodata catalyst, and elements. So HAIL is a useful computational language for high parallelization of genomic data. And HAIL is built on Python. So I found in my programming that Python can be useful to know, but a lot of things are done in R, and in biodata catalyst we do have SAS available. So it's a tough question because there are many different routes to usually get to doing a particular type of data analysis. I think HAIL is one example of it's written on top of Python, so you need Python. But in most applications I see, there are routes to doing data analysis in all different platform languages and all different languages. So if you come up through a school of public health that only taught you SAS, I remember learning how to do some pretty complex things in SAS back in the day. So they're all full-featured programming languages. So I don't know if there's a real answer other than that Python and R are probably the most common languages. I don't do Python programming, so I use ChatGPT like things if I have to do Python programming. Well, we already are working closely together. So there's some great interoperability projects right now between AnVIL and biodata catalyst. In fact, we have projects on happening right now to do analysis with AnVIL data on all of both of the biodata catalyst platforms. Some people might hear about this biodata catalyst powered by 7 Bridges and biodata catalyst powered by Terra. So we're working on ways that allow researchers to do analysis of AnVIL data on the biodata catalyst powered by 7 Bridges. So I think we have the benefit of already talking, just so that we can maintain interoperability and try to do more interoperability projects. I would love to learn more about AnVIL projects. You know, maybe I'll come back next year and show you guys what cool things we've been able to do with data aggregation across all the different platforms. There you go.